The Cannabis Conversation. A European perspective on the emerging legal cannabis industry.
Welcome to the Cannabis Conversation with Anuj Desai, where we explore the new legal cannabis industry by speaking to the professionals that are helping to shape it. So I hope you had a very good week. More great news on the cannabis front coming from New York this time. This week, the New York legislature passed a bill to legalize recreational cannabis. New York is obviously one of the biggest states in the US, and it's estimated that annual revenues could be as much as $2.5 billion in just a couple of years' time, which is obviously quite sizable. What's really nice about this story is that there's a very strong social equity theme to it, with some rules to prevent basically the big players from really dominating. So there's open competition in the state. 50% of the licenses, I think, are reserved for social and economic equity applicants. But also some of the tax revenue that is collected will be redistributed and reinvested in communities that have been disproportionately affected by the war on drugs. So this is really great news on a number of levels, and hopefully will serve as a very good model for other states and indeed other countries that are looking to pass reforms in this area. On a personal front, things have been getting busier and busier for me, which is great. Really very lucky to speak to a huge variety of people across the whole sector. And that really is just one of the best things, because just I'm constantly learning. Every day I'm learning new things. And they have said it's a key factor in quality of life is lifelong learning. So I'm very grateful for it. I am speaking to lots of entrepreneurs, and it's funny, I do still seem to speak to a lot of people who are really, really keen on starting cultivation businesses. And, I wouldn't discourage anyone, but it's I think it really is a very hard business. So I'm speaking to people across the whole value chain and really understanding that growing cannabis, particularly medical cannabis in Europe, is really very challenging from a regulatory perspective, as well as getting all of the testing regimes and the quality and consistency of the product, and fitting that all into the very strict rules around production, manufacturing and processing a medical cannabis product. I think people might not have an idea of just how hard that all is, and many, many big businesses who have been doing it for a while still struggle to maintain that. What I'm actually finding a bit more interesting are the companies that are focused on sort of generating IP and researching, formulating and producing medicines. I think that's a hugely exciting area. And I think because there's just so much work to do in this area, there's lots and lots of scope to build some really solid long-term businesses if it's done the right way. As ever, if you're in need of a UK lawyer, please do get in touch at Anuj, ANUJ, at canverse.global or via LinkedIn. Got some great clients at the moment. I'm helping them with a variety of areas such as investment fundraising, corporate M&A, and some big commercial deals. And we also are helping people with brand and data protection and day-to-day legal needs like that too. So if you have a need, please do get in touch. Now on with this week's show. Really, really interesting episode. It's a 101 on cannabinoids. Enjoy. On today's show, we have Professor Saoirse O'Sullivan and Dr Andy Yates. Saoirse and Andy have a number of roles within the medical cannabis space, but are primarily here to talk in the capacity of being advisors to Artelo Biosciences. But they can tell us a bit more about all the wonderful things they're up to in a minute. Guys, welcome. How are you?
Great, thank you.
Yeah, awesome. Thanks for having us.
Yes.
Real pleasure. I think this is a great topic, actually. We're going to talk about cannabinoids and synthetic cannabinoids, which is not something I've covered specifically, so I think there's quite a bit to talk about. To begin with, let's do the traditional thing, and let's start with a bit about yourselves. Perhaps, Saoirse, do you want to start and tell us a bit about your background and how you got into cannabis and why, and the various things you're up to now?
Yeah, okay. So I got into cannabinoid research in 2002, actually. So I moved over as a postdoctoral research. This was after my PhD. I moved to Nottingham and started looking at how cannabinoids affect the cardiovascular system. And this was a 3-year contract. And I basically got hooked on cannabinoid science during this time. I just found it so interesting. It was still really early days, and when people were discovering the cannabinoid receptors and the endogenous compounds that activate them. So it was really an exciting time to be in a novel research area. So it was only supposed to be for 3 years, and I ended up staying there for 18 years and worked my way through the ranks of academia, eventually leaving last year as full professor of pharmacology. So I spent that whole time doing research on cannabinoids. It started off very much, preclinical, doing laboratory experiments and cells and animal models. And then I worked towards human work. I did studies in healthy volunteers and clinical trials. So I really started moving towards the therapeutic uses of cannabinoids and how we can kind of best tap into that. And that led me naturally to working with the pharmaceutical industry, which is why I ended up leaving academia, was because I wanted to try and make a real difference and really try and help clinical trials get initiated, try and help companies to understand the science that was out there, what we already knew, and trying to help them match the science with a potential market. So that's what I do now, is I act as a consultant to various companies, and we're talking today about Artelo Biosciences, who I've been working with for a number of years, but that's what I'm doing at the moment now.
Brilliant. And you mentioned you got hooked on cannabinoid science. What was it that drew you to it? And particularly in 2002, very early days, were there any kind of issues with stigma and overcoming that you encountered when you decided to look into this area?
Yeah, so firstly, I'll say that I really got into it because it was just very exciting because of the novelty of it. And it was also a super friendly community to work in. So when I started going to cannabinoid science research conferences, I just really found that people were really friendly. They were very open to collaboration. Everybody just wanted to see cannabinoid science excel. And I had come from working in hypertension, so high blood pressure research, which was quite a patriarchal research area. And so it was just a real change to move into the cannabinoid community. And so it was a combination of the science and the people who worked there. That was really what kind of kept me in it. And because I could see there was just huge potential. So that was quite exciting too. As a scientist, you don't often get to tap into research area that is still in its infancy, and therefore you can really be part of the journey. So that was exciting. But yes, definitely came up against a lot of stigma over the years. So the first kind of 5-10 years trying to get research funding was incredibly difficult. Trying to overcome that barrier that there was a science behind these compounds, that there were receptors, there was an endogenous cannabinoid system, that what we were doing was real science, was difficult even within the scientific community. So it was very difficult to get funding. People just didn't take it seriously. I mean, there's still an element of that people think it's all about just sitting around and getting high. I get that kind of comment a lot when I tell people I'm a cannabinoid scientist. I don't think they quite take it as seriously as if I said I was a geneticist or something. But it's changed a lot in the last 3 to 5 years. I think there's just been a massive sea change in terms of public acceptance and acknowledgement of the potential of compounds. And so that's made life an awful lot easier and has actually really facilitated this industry moving on leaps and bounds in the last 3 to 5 years. And it's really on an upward trajectory now. If you look at the amount of research that's being done, it's crazy. I used to know all the names in cannabinoid science, and now papers are coming out and I'm like, I don't know these people. And so it has really expanded. So that's amazing to see. You know, you started with a little baby and now it's become this huge entity of itself. So
yeah,
that's what's kept me in it.
Yeah, fantastic. And yeah, I mean, it's great to have that sort of longevity of experience to sort of talk about. I mean, as I said before the show, a lot of people I speak to sort of in the last 5 years have got into it, but you're sort of nearly pushing 20 years, which is amazing. And to seeing it change so much is crazy.
Yeah. And you just have this kind of long-term memory of stuff that you've seen and read and heard and conversations that you have. And cannabinoid science and cannabinoid pharmacology is very complex. You know, we're talking about a whole plant, which is hundreds, if not thousands of molecules. When I started this in 2002, I thought it was complex. So I can't imagine what it's like for somebody coming into this area now, because it is so complex and cannabinoids work on so many different targets in our body, receptors and proteins and ion channels. And, you know, it's massively complex. So yeah, if I thought it was bad 18 years ago, I can't imagine what it's like to be a new person in this field of research.
Yeah, absolutely. Well, we'll go on to talk about cannabinoids in a second. Andy, would you mind just giving us an intro on you and your background and the various things you're up to?
Sure, I couldn't agree more with Saoirse. Thankfully, I'm not new to it either. So I was also doing research on cannabinoid science back in around year 2000 at the University of Nottingham in a lab very close to Saoirse, although I don't think we ever formally met, although I'm sure we passed on a corridor or two.
We must have passed in the tea room at some point.
We must have. And so, yeah, I got into cannabis research because when I was doing my pharmacy degree, I strangely enough studied a module, an optional module called drugs of abuse. And within drugs of abuse was the cannabis plant and how people used it. And I was always fascinated, actually, and it was put together very well, that module, just about how cannabis, if you wish, was classified as a drug of abuse and harmful, and alcohol and tobacco, which are obviously used as the standard medicinal drugs that were legal, were not classed as harmful and therefore not illegal. And that got me thinking about, well, maybe there's some interesting work to be done there. And I didn't really go into it from a sort of medicinal side of things. I just came in from it from a general interest that this drug had got this label, and yet the sort of evidence didn't sort of stack up to support where perhaps I saw it at the time. So I actually did my Ph.D. thesis on synthetic cannabinoids, which I know we're going to talk about later in this podcast. And my role at the time was to say, well, okay, all the things that go on in the head are harmful, and all the things that go on in the body may be less harmful and less, get you less high. So my research was really to see whether we could design molecules that just targeted the receptors we thought that existed in the body at the time, leaving the ones in the brain alone, and therefore being able to create a medicine. I think things have moved on massively since then. And I spent nearly 12 years in large pharma industry, mainly for AstraZeneca, working in a whole host of different roles in the drug development and commercialization cycle. And I guess to my surprise, having done a PhD on it and learned some of the wonders that I believe cannabis medicine and cannabinoids can offer, not really ever seeing large pharma take them up on a sort of big scale. I mean, you'll hear later that one of the drugs we're working with today at Artelo Biosciences came from the laboratories of AstraZeneca. But as a general rule, they didn't seem hugely interested in them. So when I left AstraZeneca in 2016, I decided that actually there was a great opportunity to bring all my pharmaceutical company experience, plus my knowledge and passion, if you wish, for cannabinoids, into the industries and businesses that were setting up in the here and now in the last 5 years. And, I really believe we've sort of fallen on our feet working for Artelo Biosciences. And obviously, Saoirse and I were there right from the beginning. So I think we've helped build the company in the way that it's fallen, because we're now working on 3 different drugs that each modulate the cannabinoid system, the body's cannabinoid system, in different ways. And of course, that for Saoirse and I with a science background is incredibly exciting, because it's more shots on goal. And obviously you can start to see right from the get-go that if they're all slightly different, then they will all have potentially different uses, different side effect profiles. And that way we can sort of pick the winners for different diseases that we'll get into from the portfolio of opportunities that we've got.
That's brilliant. And thank you. That's a really great summary of your background and how you came to be studying this area, and a very nice segue into the main topic. So we'll talk about synthetic cannabinoids in a moment, and we obviously use the word cannabinoid a lot on this show. Maybe we just start at the very beginning, and what is it that actually defines a cannabinoid, and what are the sort of defining characteristics of it? Is it simply that it's something that binds to the right receptors, or is it wider than that?
So I think the official definition, which is actually under debate within the science community, even whether this needs to be altered or not, is that a cannabinoid is anything which binds to cannabinoid receptors or is structurally similar to those chemicals which bind to cannabinoid receptors, because we know that an awful lot of the plant compounds and the compounds that we make in our body don't actually bind to CB1 and CB2. And we still call them cannabinoids or endocannabinoids. So it's a fairly broad term, but any chemical that comes from the plant or is structurally similar or binds to either the CB1 or the CB2 receptor is called a cannabinoid.
Well, the only thing I was going to add to that was that I do a bit of a sort of presentation developing cannabinoids. And of course, it's a nice title to have, the renaissance of cannabinoid research. But I get to a point where I actually just sort of talk to the people that are in the discussion that really we should actually treat, for drug development purposes, we need to think of cannabinoids as snowflakes. They all sort of look the same on mass, maybe, but actually when you get down to the microscopic level, they're all individual. And that plays out in the pharmacology, the legal status of them, country by country, how you sort of manufacture them and how you get into the body. So it is a really fascinating discussion. We sort of use this throwaway term, cannabinoid, without really actually thinking sometimes behind what's behind that word.
Yeah.
Yeah. And from a drug point of view, you have a class name like that usually implies they behave similarly. And with cannabinoids, we know that that's not true, as Andy said. They all have a very unique kind of physiological and biological effect. So lumping them under this umbrella term makes it really difficult for people to be able to kind of dissect the evidence of what they do, because they all do different things.
Yeah, and I would expect over time the sort of nomenclature will expand to sort of cater for these different types of cases.
Yeah. At the moment, it's broadly just put into endocannabinoid, phytocannabinoid, and synthetic. That's at the moment, that's currently how they're categorized. It's just by those three terms, just to define where they've come from. But that really needs much more refining.
Yeah, as you brought those up, why don't we just talk a bit about those three classes and broadly what the differences are?
Yeah, so a phytocannabinoid, as the term phyto derives from the word plant, means that it has come from the plant. That is generally cannabis sativa, although some plants do make chemicals that have some cannabinoid receptor activity. So we can't say it is unique to the cannabis plant, but it is mostly the cannabis plant that makes these compounds. And then the endocannabinoids are the compounds that we make in our body that bind to cannabinoid receptors. There's probably 8 or 10 of these different molecules, and they're just all named together under the term endocannabinoid. And then a synthetic cannabinoid is something which has been made in the laboratory. So it's not come from our body, not come from a plant, but has been produced synthetically in a laboratory to either be the same as one of these compounds, so you can make synthetic versions of a phytocannabinoid, or an endocannabinoid, or it can be a derivative, so changed slightly to alter the characteristics of that molecule. So that can make it more potent, or, just change the kind of profile of the molecule. So sometimes a synthetic is exactly the same as a phyto or an endocannabinoid, and sometimes it's structurally different.
Right. So you've sort of tweaked bits of either an endocannabinoid or a phytocannabinoid to, I assume, enhance certain characteristics.
Yeah.
So as a good example, within the Artelo portfolio, we have 2 synthetic cannabinoids where we actually have a third which modulates the endocannabinoid system. Let's talk about the 2 synthetic cannabinoids. We have a co-crystal of CBD, cannabidiol. And in order to make that co-crystal of cannabidiol, we need cannabidiol. You can either get that from a plant or you can get the chemist to make it. So they're sort of bioidentical. The chemistry is identical. We have chosen in our molecule to use synthetic because it's a cleaner profile. You don't have some of the other phytocannabinoids in there. And we just get chemists to make it. We bring it in. But we can also use phyto CBD for that. Our other molecule, ART2713, is completely different from all these chemical structures you might find in a plant. It truly has been designed in a chemist's mind, probably actually in a chemist's computer, to mimic how the atoms and the bonds look in a phyto-derived cannabinoid, but looks completely different chemically. And because of that, we can do things and design that molecule to be better, if you wish, in inverted commas, than what you can get from the plant. With nature, you're stuck with what the plant chooses to produce for you. With true synthetic chemistry, you can go away and design that molecule to do certain things that cannabinoids from the plant can't do.
Just to add to that, that can be in terms of biology, so making it a more potent compound, or it could just be changing the physical properties of the chemical, like making it more soluble, which we know is a problem with cannabinoids. So it can be because you're trying to alter the chemistry or the biology of what the plant makes.
Right. Very useful distinction. It's worth mentioning at this stage, I think, is there a terpene that is also considered to be a cannabinoid?
Not that I'm aware of. A terpene that binds to the CB1 or the CB2. Oh, are you thinking about beta-caryophyllene?
That's the one, yeah.
Yeah. So it binds to the CB2 receptor and so could be termed as a phytocannabinoid as opposed to a terpene, because it has cannabinoid receptor activity, so therefore could come under that vague class of being a cannabinoid.
Right.
And people are looking at whether it could be used, because CB2 is very good for anti-inflammatory properties. People are looking at whether that compound could be used for inflammatory conditions.
So you can see the fun we have, Anoush.
I mean, yes, so much to get your heads around.
It's mind-boggling, yeah.
Completely.
And wherever you look at the field of cannabinoids, it always strikes me that even those people that sort of write upon it at the highest levels have their own interpretation of what we've just talked about, or sometimes blatantly get it actually wrong. And I've seen that a few times in my passing, that you can see a description of something which talks in depth about how THC cannot be synthetic. And it's like, well, it can be if you make it in a lab. So
yeah,
it's complex.
Yeah, and I mean, what number are we on at the moment? Is it 144 phytocannabinoids, or is it more now?
I'm not sure that we have an exact number, but it is getting towards the 150 mark, I think, and then hundreds of other types of chemicals which aren't specifically classed as a cannabinoid. So the terpenes, the flavonoids, all the other chemical constituents of the plant.
Yeah. Fantastic.
Yeah. There's a point to be made there, which I think is worth bringing up. It's not something that Artelo is specifically researching, but as you can imagine, in a plant, there's a sort of pecking order of which cannabinoids the plant pumps out the most. And when we're getting down to these very, very minor cannabinoids, there's no way that economically you'd be able to get and extract them from the plant, but they may be very interesting from a pharmacological or biological perspective. And this is where the interplay between phyto and synthetic is so nice. So we identify one that's made, by the plant at a yield of 0.01%, but we can get the chemist to work on that, scale that up, and end up with a very pure form of this synthetic version of the phytocannabinoid of interest.
Andy, that was a beautiful segue into my next question. So as we drill down onto synthetic cannabinoids, you've mentioned a couple of reasons, but, why create them in the first place? There's obviously a big section of the cannabis community who are very passionate about the plant and its part in medicine. But clearly there are some advantages to synthetic cannabinoids as opposed to phytocannabinoids. Would you mind sort of giving us a picture on that at all?
Well, there's one really important thing that synthetics did for us that never really gets acknowledged often when people talk about it, is that the first synthetic compounds that were made were designed to bind to the CB1 receptor, and they are what led us to the discovery of the CB1 receptor. So without them, we wouldn't know, because they made radio-labeled synthetic molecules that could be used as research tools. That's why they were first made back in the 80s. And those radio-labeled molecules are what showed us that we have cannabinoid receptors in our brain. And once we found that we have these cannabinoid receptors in our brain, which were lit up with these synthetic molecules that were designed to bind to them, then we realized we must have an endogenous cannabinoid system, because why do we have these receptors if we don't make molecules in our body that bind to them? So without those first synthetic research tools that were developed in the 80s, we wouldn't know that the endocannabinoid system existed. So I just wanted to get that in there as a little flag, a little one love to synthetics.
A little hurrah
Yeah.
to the chemists out there.
And that's why they were first developed. What happened next is different, but that the first ones that were developed as research tools.
Right. Okay.
And that's how I came into the medicinal chemistry side of cannabinoids as research tools. They were just starting to find their usage in pharmaceutical companies who obviously like synthetic cannabinoids because there's intellectual property around them. If you try to patent something that comes from the plant and has been around since the 1970s in the public prior art, then you can't get as solid patents positions on those things. So pharmaceutical companies like new chemistry means that they can get intellectual property, and that means that they can protect their investment in the R&D that's required to turn those molecules into medicines, licensed medicines. And I know people don't like to talk about intellectual property and profit for pharmaceutical companies, but it really is a driving force for all of the innovation we've had in medicine in the last 50 years, the fact that somebody can protect what they invest in. So that's one reason why synthetic cannabinoids that are structurally diverse from what the plant produces are important. But of more importance from a medicinal perspective, a medical perspective, is that you can do things with these molecules that you can't do with a plant-derived cannabinoid, such as Saoirse said, ART2713 has been designed so that it's more soluble and therefore more bioavailable. We can give a very small capsule reliably and consistently to a patient, and they absorb that cannabinoid, whether they're fed or fasted, whatever their sort of situation is, they get the same dose of the cannabinoid. And that's because we've changed the properties of the molecule. When it goes into the body, we've done some other things to the drug. THC, for instance, most of your listeners will be familiar with THC, is known as a partial agonist to the cannabinoid receptors. That means, in the terms, I'm not a professor like Saoirse, so for me it's dimmer switch time.
That's my analogy.
It's not mine too.
I started it.
So we have I've borrowed it. It's a good analogy for me to use. So no matter how much THC you take, you'll only ever switch the lights 3/4 on with that sort of dimmer switch analogy. With the drugs we've created, you can take those lights up to 100% and actually up to 110, 120% in some cases. Now, why might you want to do that? Well, you might want to do that if you get to see that some of the effects of your cannabinoid are different from THC, because you'd expect there might be a difference because we're a full agonist and THC is a partial agonist. Obviously, you've got to watch for side effects as well. It might be that there's more side effects, but ran in a controlled, randomized clinical trial, all of that can be worked out. We've obviously seen some data with ART2713 with its ability to put weight onto patients that we're perhaps a little surprised with. You wouldn't perhaps get to see that with what we've seen from the literature around partial agonists. So our thinking is maybe something to do with the way it works pharmacologically. The second thing we've done with ART2713 is we've made the drug less able to pass through the blood-brain barrier. So the blood-brain barrier is, as the name suggests, the barrier that separates the brain from the peripheral body. And what we can do by putting in some chemistry that stops the molecule freely floating past that blood-brain barrier is we selectively keep the drug in the body and acting on the peripheral receptors. Why might we want to do that? Because we know that a lot of the CNS effects of cannabinoids are generated in the brain. And therefore, if you can stop it from getting into the brain, your highs, your euphorias, any sort of psychosis that you might expect, all of them will be lessened because we're getting the drug to do the job at the site where we want it to, which is in the body. So all of that can be achieved through synthetic cannabinoids that just can't be done in the same way with plant-derived.
Thank you. That's, really good explanation and again, great to get that context the benefits, I suppose, of synthetics over phytocannabinoids. Kind of follow-on question to this is, how do you actually go about creating these then? I've read about biosynthetics created from yeast and things like that. Are these some of the methods that you use, or what are the kind of main methods that people use to create these in the lab?
Yeah, so you can do it in different ways. Obviously, if you're trying to recreate the phytocannabinoid, so let's say we want to make THC, first of all, chemists will elucidate the structure of THC, and this was done, Saoirse can tell us, in
the 1940s. 1940s for THC, yeah. And then infamously the 60s is when it was further done, but the 40s is when the first chemical structure was elucidated.
There we go. So you've got this structure, you literally got the building blocks, I think of it as Lego, so you sort of know how the Lego's mapped out. So in that case, you'd give the job to the chemist to say, build me an identical one. And then they'd get their little bits of Lego and they would use their chemistry and stick it all together. And the two would be absolutely identical. Couldn't tell the difference. So that's how you mirror making a synthetic bio-identical version of a plant-derived. But then the next obvious thing is that if on THC you've got a methyl group, which is a carbon and 3 hydrogens, well, the next logical, the way a chemist brain says is, well, what happens if I turn that into an ethyl group? That's 2 carbons, a propyl group, 3 carbons, and you start to build off what is the original phyto-derived cannabinoid, and then you hand it to someone like Saoirse and you say, what's happened to the pharmacology? And you'll get reports back that, less binding on CB1, more selective for CB2. So we track things. And at a certain point, we probably have enough information to use what's known as SAR. So that's called structural activity relationship. So we can start to work out that if we stick big groups up here, at the north of the molecule, that sort of lessens CB1 binding. And if we change that to sticking it on the south of the molecule, we increase CB1 binding. And so we can rationally choose what our next chemical steps are to optimize the molecule to whatever we want to do. Then the molecule of interest most to me, which is ART2713, which is the one we're going into the clinic with at Artelo Biosciences. This doesn't look anything like THC. You wouldn't be able to identify it in a police lineup that it looks anything like THC. And what they've done there is that chemists have then done using a computer, they've sort of sucked the elements of THC into a computer, and they've then used in silico modeling. So they've sort of mapped on different chemistry, but biologically it looks the same. So to the receptor it looks the same, to our eyes it looks completely different. And again, they've done that for the reasons we just mentioned before around developing a better molecule. And that's what we've done. So we've got what's called a benzimidazole structure in ours, different ring system altogether. And we can sort of literally decorate the molecule all around the outside of it with different chemical groups or moieties. And again, we went through that same, AstraZeneca on our behalf, because they did some of that hard work in the labs, and then we licensed the molecule from them. They went through this rational drug design and ended up with a structure that looks nothing like THC, nothing like CBD, and yet hits the CB1 and the CB2 receptor really hard as full agonists, very potent. And we now make that at a chemical factory, very high purity, 99.9% purity. And we make it in about 8 chemical steps, adding A to B to make C, and then we add some D to that, and we build it up.
Wow. Well, the thing that really struck me when you were describing that was just how many options you have when you're sort of tinkering around at that beginning stage, which helps to explain why these things take so long, whenever, particularly relevant with COVID, but why it takes so long to develop drugs when you take 10 years or whatever, and all of the preclinical trials that you have to do. You find the right bit, the right methyl or ethyl group to stick on it, and then you've got 1,000 places you could stick on it.
And then you've got to match all of that to the biology as well. So at the same time, you're doing experiments to try and figure out what is tweaking that molecular structure doing to my biology? Is it making it better or worse? And so you're always matching that up so that eventually you end up with a molecule that seems to be perfect for the job that you want and gets all of the benefit with as little side effects as possible. So that's why drug discovery is very long and very expensive.
Yes. Yeah. No. I mean, makes complete sense. Are there any kind of particular challenges to synthetic cannabinoids or this in general? It's a difficult question to ask, I guess, in relation to phytocannabinoids.
I wouldn't say challenges. I mean, here in the UK, when I was doing my PhD back in 2000, a lot of the synthetic cannabinoids were really good for us to use in the research world because they weren't controlled drugs. They weren't THC, which was a Schedule 1 controlled drug. So we were happily making them in the laboratory as pharmacological tools, future drugs, etc. And the one thing I think that's probably worth bringing to bear on this webcast and for your listeners is that then, I don't know when it was, but maybe around 2010, that this sort of substance spice came onto the scene, which were synthetic cannabinoids designed to get around the Misuse of Drugs Act. And chemists were making all those changes we just talked about, but they were doing it in an uncontrolled way. And the first person to test them wasn't a cell system, and it wasn't an animal model. It was humans on the street. And as you can imagine, there were some pretty nasty effects of these. No one had any idea what the dosing was. And they've got then this sort of bad reputation since that time, and the government sort of cracked down on them a lot more. And so one of the sort of issues with synthetic cannabinoids, I think, is a practical issue, is that nowadays they're sort of seen perhaps through a different set of glasses, which is, well, these aren't very good, are they? And they, the Home Office has started to look at them with much more scrutiny. So they're not as easy to work with as they once were from a sort of controlled drugs perspective. Even though I've just told you that our drug doesn't really pass the blood-brain barrier, it doesn't really get you high. But yeah, it now falls into that bucket. So that's one of the things that's challenging about them.
Yeah, I would just totally agree with that. I think that people hear the word synthetic and cannabinoid together and they automatically go to spice type molecules, and that you're messing with the plant molecules in a bad way, as opposed to the fact that synthetic just means you have produced it in a lab. And I think that word synthetic, the meaning of it, and especially for patients and the public perception, has altered a lot because of spice compounds. So now there's a stigma when you work in synthetic cannabinoid science. There's a stigma, like we used to have a stigma before 20 years ago. We have a new stigma, which is getting over this concept that synthetic equals bad. And it doesn't. It just means made in a lab. Yes, there are bad synthetic compounds, but there's an awful lot of really good synthetic compounds as well that are being developed in drug discovery. And I think we need to work quite hard to really dispel that idea that synthetic equals bad.
Yeah. I guess it's a problem that cannabis has in a number of respects, where everything's reduced to good or bad, and there isn't the sort of nuance. Yeah. Everything is skunk and it's terrible for you, or
exactly
all of these things, without kind of fully appreciating just the huge wealth of different things inside the plant.
Yeah.
And the other thing as well is, I would like to get this over to any of your listeners, that do come from the cannabis plant is natural. Natural's good for me. And without doubt, for some people, that's the case. That's their mindset. That's their belief system. But what I can reassure everyone of is that in order to make a medicine, as Artelo are doing, and in order to get that medicine licensed by the MHRA and the EMEA and FDA, depending on where you live, you have to go through so many hoops to show that it works, that the side effect profile is beneficial against the effect you're looking to have. So the benefit risk ratio is right, that you make it to a high purity every time, and every time a patient takes it, the same dose goes into them. So all this quality is just built into a medicine right from the beginning. And of course, in the case of a lot of plant-derived cannabis, that's just not true, because the money isn't being spent on them to do all of this evaluation. It's a bit like, as I said before, even with the plant-derived, often the first people to get the new formulation or the new strain of the plant is the patient. It's not gone into any cellular models, and it's not gone into any animal models first to establish itself. It's just assumed it must be okay for humans. So I think for me, you mentioned issues with synthetic. I think there'll always be a group of individuals in the cannabis community that will see synthetics as bad, that they want the natural stuff in the plant. That can't harm me. I mean, I come from the school of pharmacy, literally, where you know that there's plants called deadly nightshade for a reason. That not all things that are contained in plants are good for you.
I was just going to add to that there are some conditions where it's really important that we're not kind of tinkering, that we have a very firm idea of what chemical is needed, what molecule is needed, at what dose. That can only be done through these kind of programs. So there are conditions for which you really want to have all of that information up front before you go into a patient, rather than maybe some where there's a bit of tolerability for being able to kind of tinker around with different strains or doses. But there are some conditions and some really vulnerable patient groups where I don't think that's appropriate.
Yeah. Yeah, and it's really, really useful to add that context to it. I went to a talk by Professor Dedi Mary where he was saying, he often comes up against this in this sort of natural versus synthetic kind of argument. And, and people say, like, it's a plant. And I'm going to make this number up, but I'm pretty sure he said something like, 70% of plants are poisonous. And, so it's not always, and deadly nightshade is a good example of that. So I think it's good to offer that balance.
Another thing that perhaps just to give you your listeners another perspective, and it's not to knock at all cannabis medicines. I mean,
because we're all massive enthusiasts.
Yeah, other hats that Saoirse and I work on is helping patients get hold of good quality, high quality, ethical, legal cannabis medicines, as in extracted from the plant. I'm not a knocker at all. I just feel blessed that I can wear both hats. But just to give you an example, when we're working with our synthetic cannabinoid Art2713, which is going into cancer patients who have anorexia with a hope of producing weight gain on them, giving them their quality of life back. When we control for that medicine, when it comes out the lab and we put our specification on it, it needs to be greater than 99%. Now, that other 1% of stuff, we also need to know what it is and how much of it's there. And we need to what they call qualify it. Now, when you're thinking about a cannabis extract, sometimes you might be lucky to get 85% of THC or CBD. And the other 15% is seen as the entourage effect. That's the other stuff. We would call that in the pharmaceutical business contamination. That's contaminate. That's the other stuff.
That's the unknown.
Yeah. Now, unless you can control for that, and that contamination or that entourage effect is the same every time, and you can guarantee it's the same every time, then really, actually, every time you take a cannabis extract, then you potentially will get a different effect because you're not controlling for what the other stuff is. I just wanted to bring that in because, it's another thing that we massively control for. The MHRA inspects the plants where we make our drug, so that they know it's made consistently, safely, ethically every time. And this doesn't always happen with other plant-derived cannabinoids.
Absolutely. And actually, I'm just conscious of time, so I think we need to wrap up now. But that whole idea around entourage and polypharmacy, I wanted to go into. So perhaps we can maybe have a follow-up, because I think it's a fascinating area.
It is a whole massive thing in itself. I mean, I could literally talk all day about entourage. It is
brilliant.
Yeah, it's a massive topic, and it's a very interesting hypothesis.
Well, I mean, that's perfect. Then that gives me another show idea, which I always love. So let's get you guys back on and do that. But for the time being, I'd like to say thank you for joining me today. It's really, really interesting. And obviously we only just scratched the surface, but I think it'd be really, really valuable for the listeners to hear this. So thank you.
You're very welcome. It's been a really good chat.
Likewise. Take care.
I feel like we could talk all day.
Yeah, and hopefully it will be informative, to your viewers, so that they can go away and form their own perspectives and views on these important matters.
Absolutely. And I'm looking forward to part 2 already. So yes, brilliant. Thanks, guys. Have a lovely day.
No problem. Lovely to meet you. Bye.
Cheers. Thank you.
Cheers.